I'm delighted to be able to introduce Dr. Stephen Chanock as today's speaker. Stephen is the director of the National Cancer Institute Division of Cancer Epidemiology and Genetics, and is a world-leading expert in cancer genetics and epidemiology. Today, Stephen is going to be talking about some of his fascinating recent research, which was published in Science, on the effects of radiation using data from individuals in the vicinity of the Chernobyl accident. If you have any questions for Stephen, please either post them in the chat or raise your hand at the end of the talk. And after that, please start whenever you're ready, Stephen.
Well, thank you. It's indeed a pleasure to be here. I appreciate the invitation. Thank you, Lawrence, and thank you, George and company. I have nothing to disclose other than I will not mention Mendelian randomization in the next hour, which is unusual for a lecture in Bristol, but I'll leave it at that. I'm going to talk about using genomics to investigate radiation-related issues following the Chernobyl accident. I'm going to tell two different stories. All right. We all are aware of the tragedy of April 26, 1986, the explosion in reactor 4, which resulted in an open-air reactor core fire that was contained 9 days later, with a tremendous amount of radioactivity spewed basically around the Belarus, Ukraine, and Russian environment. And the reason this is so important for someone who's studying cancer is we know that ionizing radiation is a strong and potent inducer of cancer. It's a class 1 IR carcinogen, well established, and it's established risk factor for multiple cancers, particularly hematopoietic cancers and thyroid cancers, but we also know about therapeutic radiation where there's intense exposure. So here, one of the themes of this talk is that the type and the extent of the exposure really is different than what we've seen normally in individuals who have therapeutic radiation who develop a second cancer in the field of radiation, for instance, or individuals who receive I-131, iodine 131 for a thyroid ablation due to a thyroid condition. Animal studies have been very, very extensively pursued with, again, intense exposure showing double-strand DNA breakage. So when we look at the map here, you can see the yellow star of where the reactor was, and Pripyat is the closest town. Not that far from Kiev. It's kind of scary. The weather patterns saved a major area like Kiev from major exposure. But as you can see, it spread a whole set of isotopes across particularly southern Belarus, northern Ukraine, and some of it blowing towards the Russian Federation, as you can see here. And there are really two different types of exposure. One is the residential exposure from fallout, and particularly I-131 is what has been studied most extensively, because that's what goes into the environment, into the grass. The cows would drink it, and then the milk that the children would consume would have I-131 that would be concentrated in the thyroid. And then there's the occupational exposure of I-131, but certainly other radioisotopes for the so-called liquidators, the people that were brought in to clean up. And there are two sets of cohorts that we've been very engaged. So Chernobyl health effects, the National Cancer Institute has had long-standing interest from Gil Beebe and Elaine Ron were pursuing this in the late 90s and the early aughties, and then Maureen Hatch, and then our esteemed colleagues, academician Tronco and academician Bazika, who each direct two institutes, separate institutes. The IEM is the Institute of Endocrinology that does thyroid surgery and analyses with Professor Bogdanova, and then Professor Bazika oversees the NCRCM, which oversees NRCRM, excuse me, that oversees the liquidator cohort, in which the second study. And then we here at the NCI have a strong, long-standing commitment to Chernobyl and looking at the sets of cohorts and kinds of studies from radiation the exposure to the dosimeters, the first two individuals to the epidemiologists, particularly Lindsay Morton and Amy Barrington, who've been instrumental in the stories that I'm going to tell. So the first story is the environmental radiation exposure and genomic alterations in papillary thyroid carcinoma following the Chernobyl accident. So papillary thyroid cancer is a well-established cancer with a much higher incidence in women, with roughly a 95% survival in the UK and in the US. Cancer of the younger adults. It's highly differentiated. There's a lot of pathologic discussion, not in terms of bad pathology, interpersonal pathology, but the pathologic classification as to what needs to be treated and when things are detected by overdiagnosis with the use of more sensitive ultrasound and the like have spawned a major debate about how and in what ways to approach papillary thyroid cancer. The mutational burden is higher in older patients. So if you look at the mutations, such as in the Cancer Genome Atlas, which I'll talk about in a little bit, and some of the mutations actually have prognostic implications, such as the BRAF 600 and the TERT promoter mutations. They can drive therapeutic decisions, particularly in the UK and the United States. So we learned a fair amount about the molecular landscape of papillary thyroid carcinoma back in 2014 as one of the first cancers with a landscape analysis, in other words, integrated analysis of RNA, exome sequencing, not whole genome, and SNP chip and methylation chip, and the integrated genomic characterization of just shy of 500 papillary thyroid cancers, just about all of them sporadic. So none of these had clear clinical evidence of having received radiation therapy beforehand, and that's important. When we look at the so-called commute plot, and this is the standard that's used in the ICGC, the International Cancer Genome Consortium, or in TCGA, of giving you the landscape overview of the somatic alterations. So you can see the mutational burden. Here, the point mutations in group with C are a high fraction, almost roughly 70+% are single point mutations in just a handful of genes, such as BRAF and NRAS, HRAS, KRAS. And then there's a fraction of 20% that are fusions, that are two genes that are brought together, and we'll talk about that. And then the copy number alteration. So this is a relatively quiet tumor per se. So if we look at this, we can make certain inferences about the pathology and, more importantly, how they came about. But here, what I'm going to talk about in the next 20 minutes or so about the papillary thyroid following Chernobyl is really this extraordinary opportunity where we know exquisitely an awful lot of detail about the exposure of each and every individual who we're studying. And it was a discrete exposure. So in the past, we know that exposures in somatic mutations have been very poorly tracked. TCGA and ICGC paid virtually no attention to these exposures and environmental drivers. We're able to see with lung adenocarcinoma and melanoma, yes, smoking and UV light were very important risk factors, and we could reconstruct that they had extensive exposure. And then you could go at cosmic signatures for smoking and the like, and that's fine. But it was at a great price to work backwards, and it's been incomplete. Mutational signatures have been very useful for our capacity to be able to use sort of forensically, like some of the crime scene television shows, to work backwards in this balance between the endogenous and exogenous processes. The endogenous, those things that are quote unquote the background errors in particular tissues, which we know are different, partly based on their exposures, partly as to whether they're sanctuary tissues like thyroid, and then the exogenous processes. And we can map to select exposures, but not that many. So one of the lessons that I hope comes out of this story is never forget the context of cancer genome analyses. I think trying to capture some degree of epidemiologic interest information is absolutely essential to be able to interpret cancer genome analysis. So here we are looking at children. Our study is exclusively based on individuals who are under the age of 18 at the time of the accident, who had known exposure to I-131 over a matter of weeks to months due to their the food sources, so to speak, as well as the environmental exposure. We know that I-131 exposure has a higher risk for radiation-induced cancers, particularly of the thyroid. And a number of studies are shown here. And what's interesting is that there's a dose response, and then there's a cap to that, so to speak. When you look at it, and you cross that with age, age is a very important variable in this as well. The younger an individual is at the time of exposure, the greater the risk of developing papillary thyroid carcinoma. So here's sort of the cartoon of how we see what I'm going to talk about, from the Chernobyl accident to the weather patterns, to the environment, to the concentration in cow's milk, to the young children. Remember, this is 1986. Chernobyl took place. The Soviet Union was still in its older state, where food sources and choice was extremely limited. Very few things were available for basic sustenance, in particularly rural part of the Ukraine. And so just about all children were dependent upon milk sources for a major part of their nutrition. So let's look at the age of the diagnosis. So 1986, the event takes place. We have a number of children who were diagnosed. You can see the age at thyroid cancer, and I'll show you the dose distribution in a second. They could be up to 25 years after the accident, but everybody was under the age of 18 that we were studying. The mean age was 29.7 years, and you can see the doses were anywhere from 1 milligray to up to 9 gray. We did have access to 81 individuals who were born after 1986 who came from regions that were just south of Kiev that had no known real exposure for comparison, and that's a very useful and important group for our analyses. So you can see heavy on the younger individuals who had lower exposure, and we recognize that some fraction of these individuals did not have radiation-driven cancers, in that the lower the dose, the lower the risk. And we'll talk about this POC, so to speak, because we do know there is a very rare, but a background rate of papillary thyroid cancer. But the older, the higher doses clearly are strongly associated with much greater risk. So the age by dose, you can see here that the age at diagnosis was basically, there was no real substantial statistical significant difference between those who were in the Chernobyl region. Those who we identified from the other region, not far, that I'm not showing you the PCAs, but in terms of population genetics, look quite similar, from rural areas per se, were a little bit younger. And there was no way to overcome that, because the chaos of the time was such that no one was thinking about collecting quote unquote kind of comparison per se. So our primary statistical analysis is, this is the first time in our knowledge really being able to look at a large landscape analysis, have the epidemiology in place and the genetics and the exposure. And so we wanted to ask this question of relationship of radiation dose and PTC, the papillary thyroid carcinoma molecular characteristics. So we had multivariable regression models adjusted for age, PTC, and sex. We truncated doses at 1 gray to reduce the influence of outliers, estimated additional events above background per 100 milligray. And this is a standard way that the radiation epidemiologists approach things. And we looked at continuous, discrete counts, dichotomous categories, you name it. We spent an awful lot of time analyzing this data. And we used the Bonferroni threshold for the 68 primary tests that we did to start with. We did secondary analyses looking at radiation-associated characteristics, sensitivity analysis restricted to different exposures and with low dose to try and address this question of hormesis, which is a very challenging and controversial question, that a little bit of radiation may actually be good for you. Many of us don't subscribe to that, but again, we'll talk about that a little bit later. And the question of evaluation of departure from a linear dose-response relationship. Okay, so the other issue is, I'm not going to show you, but we did everything against the known dosimetry, in other words, the radiation exposure per individual. There is another metric, so-called probability of causation, that looks at the inferential weight to be attached to the conclusion that a particular case of cancer was caused. This is a program that had been developed by the epidemiologists here in the NCI and others over years, and is used by the nuclear regulatory industry worldwide in looking at radiation risk as a probability of causation. And our results are pretty much spot on. And I'm not going to show those figures, but they're buried in our supplements of our science paper. So here's the flow of data. We went to the Chernobyl Tissue Bank, which is a very important resource run by Dame Professor Jerry Thomas at Imperial College, who we collaborate with. We had 440 confirmed cases of PTC, all reviewed by at least 4 to 6 different pathologists who were part of the advisory to the Chernobyl Tissue Bank. We had fresh frozen material, and then we had 431 with matched normal tissue, either non-tumor thyroid tissue and or blood. And that is the non-tumor thyroid we're looking at intensely. We did some preliminary work, but we didn't publish that due to some discussions with the reviewers. And now that's something that we're looking at, this question of a kind of field effect. Is it there or not? You know, something that's very, very interesting. And we know our colleagues at the Sanger, Peter and Inigo and Mike and company are all over this very interesting question for a number of different tissues. So tissue samples were QC, DNA, RNA isolated, sequencing, expression, chips for epigenetics and copy number, genomic analysis, but then we were able to do an integrative analysis because we know the dosimetric exposure for each and every one of the cases that we had, which really allowed us to have an integrated analysis using SNP chip, methylation chip, microRNA, RNA-seq, and whole genome sequencing, looking at tumor and germline. And so I'm going to talk to you about the results of our paper and some further things as we continue, obviously, working on this very important situation. So here is the commute plot. This is a very unusual commute plot in that if you look at this top line up here, we are actually able to organize this by exposure. So in other words, there are 81 unexposed individuals, and then there are a larger group who had 1 to 99 milligray, all the way up to greater than 500. So you can look at the single base, the mutation counts are relatively low. The SBS, the single base signatures, I'll talk about that briefly, as well as the insertion deletions and the structural variants. You can see all of those color coded, but what's really striking is the driver mutation. I talked about the TCGA, which had an average age well into the high 40s, or actually a little bit more than that. And here we could see those individuals that had no exposure or very, very low exposure, again, had a predominance of the point mutations in the BRAF, NRAS, HRAS, and KRAS. But when you come over here and you look at those individuals that had higher dose exposure, 500 or more milligray, you had a much lower fraction of the individuals who actually had point mutations. And the fusion protein, the oncofusion drivers, increased in their proportion per se. And they are in a relatively small number of genes that are involved. This is a very parsimonious tumor. And I'll come back to this theme over and over. So if we look at the distribution of mutational burden in cancers, goes back to Mike Lawrence's paper some 8 years ago. And we clearly can see things like adult TCGA, where you had basically 1 mutation every 1 million base. And then you have things like smoking and UV-driven lung cancer and melanoma. You could see 2 to 3 orders of magnitude more mutations, partly because the environmental exposures were continuing for 20 to 30 years, be continually bombarding and superimposed on the development of those cancers. Here, our Chernobyl thyroid, which was in even younger individuals, was about 1/4 of what we saw in the adult TCGA. So this is more like these very efficient parsimonious pediatric cancers, rhabdomyosarcoma, and some of the pediatric sarcomas per se that have one single driver that are very efficient and have very few subclonal events. So when we started to look at the signature analysis, so we had the landscape of all these single base changes that we could see in our 440 cases. We know that there's 6 possible base substitutions. The cosmic catalog is immeasurably important for this. We could see that there are 96 possible combinations. And this is just an example of the SBS4, which is the type of mutation that you see in individuals who are heavy tobacco users. So the tobacco smoking gives the SBS4 compared to some of these other ones. 1 in 5 are your background clock signatures. So when we looked at ours, and we saw that we had 7 signatures seen similar to pediatric cancer, and 70% were due to these so-called clock, which are just the background events that continue to go on in all cancers. It's alluring to ask the question, gee, could there be background alpha or beta radiation that may be responsible for that? That's a bit of a far-fetched, but an intriguing hypothesis that we certainly considered during this. The SBS8 is an unknown signature, but when we look at this and we rest this against radiation, there was no relationship with radiation whatsoever. And we could also see this in TCGA and unrelated individuals who had sporadic papillary thyroid cancer. Very little in the way of APOBEC activity, which had been also described in the reactive oxygen, really not there. So we conclude that we could not identify a single base signature that correlated with radiation. So we then went and looked at the next sort of level, which is the small insertions and deletions, which Cosmic has been very eloquently characterizing. And there's one ID8, which is a very particular type of insertion, particularly deletions of particular size with greater than 1 base pair deletion that repeats, and lots of microhomology. Oh, I see spelling error, I got it correct. We could clearly see that this was there. And this was something that when we looked at the effect of radiation on small deletions here, we could see a very strong effect against radiation, 10 to the -9. And when we actually, so to speak, normalize this against the background of the S&V ratio, since we knew that 70+% of the SBSs, the single base, were clock, we could see that the effect was even stronger. So increasing radiation leads to more small deletions and a predominance of the ID8 signature. This is something that Sam Bajati and Peter Campbell had presented anecdotally in 12 tumors that were characterized in individuals who had second cancers after intensive radiation therapy, seeing deletions more than insertions at the microhomology junctions. And so this was very, very intriguing. And a couple of weeks after our paper was published, Flores and Roll published a very interesting paper looking at radiotherapy associated with the deletion signature that contributes to poor outcome, and indeed signature 8. What I want to really emphasize is in each of these 2 papers, you're looking at bursts of high dose, multiple gray radiation for therapeutic purposes that were given in as short and an intense fashion as possible. Whereas we, in our study, are looking at children who had weeks to months of exposure to I-131 environmentally that was steadily exposed in food sources and environment as opposed to beams of radiation therapy. We had already started down this road with a collaboration with Yuri Nikiforov in Pittsburgh, who was looking at the relationship between radiation dose and gene mutations. And we'd reported in a very small number, 65, 49 of whom overlap with our study, and all of them were exactly, we confirmed them with the larger whole genome sequencing, that there were indeed more fusions than there were point mutations in individuals with higher doses. And this is a bit of a biased set, but it certainly raised this question. As you can see, the fusions involved a very small set of genes, RET, RAF, and NTRK1 almost exclusively. And this was a very interesting observation in our mind. So when we looked at ours, we could identify candidate drivers in 98.4% of the tumors. That's really phenomenal. Other than Ewing sarcoma and a few other sarcomas where they're pathognomonic translocations, this really is unheard of, okay? To see this degree of parsimony. So we could see a set of mutation drivers and then fusion drivers as well. Then, and interestingly enough, almost 94% of them map to one particular pathway, the mitogen activated protein kinase pathway. And so this was really, again, quite striking, raising a very important question about the developmental nature of thyroid cancer, which we know the thyroid is a sanctuary cell, so to speak, and particularly the papillary regions are quite protected. So when we looked at this, we also went back and said, all right, we know I'd mentioned the TERT promoter, a very important gene on chromosome 5, had been seen in 20% of TCGA older cases. We saw it in 1 out of 386 cases in Chernobyl. And so is this an age-related phenomenon? Quite likely that that's part of it, but it was really quite striking to see that difference in the character of, or at least the distribution of drivers of that cancer. We could also look at the thyroglobulin, which is frequently mutated in thyroid tissue, 309 of the cases, but it was almost always in the introns, and we really didn't see what we considered to be major drivers of the cancer in the thyroglobulin gene. And this is really taking on quite a bit of momentum in trying to understand the development of particularly many of the adult cancers, these lineage and driven states from stem cellness, so to speak, or the aberration of stem cellness. And so the thyroid cancer certainly looks like that. And we know we have virtually no subclonality. So when we looked at the PTC drivers, the shift to fusions was very important. We could see that it increased with greater risk, with greater radiation dose, that the effect per 100 milligray clearly increased for the likelihood of having a fusion driver. The same for the indels and for structural variants, which together we could put these together to start to ask a very important mechanistic question. So we know structural variants are large events that take place that can be reciprocal inversions, deletions, they can be simple, or they can be very complex, like with chromothripsis and chromoplexy, which we saw virtually none in our particular study. So when we looked at the increased dose associated with the increased number of simple balanced structural, we could again see that radiation strongly correlated with that. And in fact, was strongly associated, not correlated, was strongly associated with that. So it gave us this hint that there's something about the breaks that are taking place due to radiation. So when we look closer at the DNA double-strand break repair mechanism, we went to some of the experts at UNC and NCI, talked to them. There are very, very interesting signatures when you actually look at the sequence. So we had the RNA fusions, as well as the RNA simple balanced translocations. And then we could go look at the double-strand DNA and see that there was a particular pattern that really fit the non-homologous end joining. So these distinct repair mechanisms. So here, when we looked at things that were greater than 5 base pair deletions, which was a very good fraction, we could see the degree of microhomology, and whether it was 0 to 1 base or greater than 2 base pairs. So here we're able to use exposure, or we're able to use genomics, and we're able to use time, and then the sequence itself to really, I think, make very important insights into actually how the cancer developed. This is an age-old question of how radiation works. The animal models have suggested non-homologous end joining, but no one had ever really seen this in humans, particularly those who had protracted lower dose exposures over a period of time. So our conclusion is non-homologous end joining is the primary DNA double-strand break repair mechanism. So when we look at the somatic copy number alterations, some of them can be driven by this. We could see very little in the way of activity other than the Q arm of 22. And when we looked at these, we could see that there were just shy of 20% in both the TCGA and in our Chernobyl studies. But what we saw was strongly associated with the driver. So if there was a RAS mutation, over 50% of them, over 50% of the time that you had a 22q, there was a RAS mutation, but rarely a BRAF. So it's telling us that there are different characteristics to these drivers per se. So we also could see the radiation effects vary by age and exposure. So the stronger the radiation effect was seen at a younger age per se. And this is something that we are now pursuing with roughly the same size, about 450, 500 further cases that we have with formalin-fixed paraffin embedded, to be able to really address this question of the importance of age against exposure and the age at exposure per se. So really, it's a very interesting question. When we then looked at the germline variation, we saw that there were no variants associated with radiation. We looked at the roughly 125 cancer predisposition genes. We looked at radiation sensitive genes that had been reported. We saw no significant increase in the number of pathogenic mutations by mutations or by genes. And we saw real insufficient evidence for gene environment interaction, which at the germline level, there was always this hypothesis, and people had written that after Chernobyl that there were probably genetic predisposition for those who were going to develop papillary thyroid cancer. We are yet to see that. Now, we don't have that large a sample set, but we clearly did not see that. And as we go further with the next 500, hopefully, we will continue to address this. But at the moment, we don't see smoking guns for highly penetrant germline variation. But when we look at common germline variation, we saw this interesting inverse relationship with a polygenic risk score. So there have been polygenic risk scores based on 12 previously reported thyroid cancer risk SNPs. The Icelanders first did it. Many people have done this to be able to make various kinds of contentions per se. And the hypothesis is that could the genetic burden be important for sporadic? And in fact, that's what we saw. So in other words, the higher the dose of exposure, the lesser the PRS, so to speak. And so our interpretation of this is that the radiation probably overcomes any and all effects of these small cumulative PRSs that you get with lots of small SNP effects, as opposed to the those individuals who we knew had no exposure, who had higher PRSs. So it's an interesting and alluring hypothesis. We haven't closed the door on that, but it certainly raises an interesting question. So we looked at the methylation patterns in the Chernobyl thyroid cancer, and they are driven by the mutation. We saw no radiation exposure. We spent months trying to line up the epigenetics against what had been suggested to be radiation type of events. We could not see that. The minute the tumors appear to develop due to either a BRAF or a MAP kinase type of mutation, that's what seems to be an important aspect. It's highly differentiated CpG island probes discriminate between BRAF and RAS-like tumors. Interestingly enough, we saw the exact same thing when we looked at mRNA clustering, okay? So the radiation dose, we saw nothing that's separated by radiation dose in our mRNA. We went to the large GEC database in the Broad and kept plugging our data in, and we could not find the detritus of RNA expression. It's the driver that had this very strong effect per se. So these were similar results for microRNA and methylation. And there had been this one gene that a German group has made a whole cottage industry of, the CLIP2. We had many more cases than they'd ever looked at in the Chernobyl tissue bank, and many of them overlapping of their set with ours. And we could not see that we could identify a particular expression pattern in this gene that one could say, aha, this is radiation-induced injury versus not. Whether you did it by age or whether you did it by copy number. This happens to be, CLIP2 is a very interesting gene right next to a gene that I worked on, cloned 25 years ago. The NCF2 is part of the CGD complex. It was one of these sort of strange things 25 years later to be looking at a region that I had spent a good part of my life looking at. And for the life of me, we could not come up with a way to look at our data and find a relationship between CLIP2 and that expression. So to tie up this part of the talk, we have this paper that's in Science that really talks about the exposure of the radiation in the environment, particularly through MELT, the double-strand DNA breaks that led to thyroid cancer in the genomic landscape, and we could see the DNA double-strand breaks. So our key findings of MAP kinase pathway is really crucial, increase in double-strand breaks, non-homologous end joining, and the epigenetic and gene expression profiles were determined by cancer driver mutations. So as I mentioned, we're going deeper into expanding the dose range for the higher dose, increasing the ability to quantify the effects of age at exposure. We have multifocal PTCs now that we're looking at, asking the question, are they synchronous or asynchronous? 46 metastatic tumors. We're just getting ready to submit this this paper, we hope this summer, and we don't see anything unusual about those metastases. They all have the exact same driver that the primary tumor has. They have a few attributes that have been described in other metastases, but by and large, there's nothing about radiation that, in other words, is developing asynchronous types of cancers. And that's where our multifocal and metastatic tumors are raising this question. If indeed our hypothesis is that you're having the bombardment of ionizing radiation to the DNA, do you get cuts in several different places that then may have their own temporal uncoordinated development of papillary thyroid cancer? And so far, we have seen very little evidence of that. There may be one or two cases of that, but that's something that we're pursuing right now. Preliminary findings of no field effect, as I mentioned before, and then the additional analysis of age at exposure as well as age at diagnosis, and of course being able to look at other tumor types and populations. This is work that really Lindsay Morton and Danielle Cariotti and Eric Dawson have been marvelous with our academician Trunco, and certainly our close collaboration with the Broad with Gady Getz and Chip Stewart. So let me just now transition for the next 15 minutes to a different question that we wanted to ask, a very complicated one that has tremendous political implications, and that is, could there be a transgenerational effect following the Chernobyl accident? This has been on the minds of people in Chernobyl, in Fukushima, and in other places. So we designed a family study. I did this 8.5 years ago with Dmitry Bazika, where we recruited families where mother and or father had been in the liquidator cohorts. They left, and then they started a family more than 46 weeks after they left. And so the first analysis that I'm going to present to you is 105 families with 130 adult children. We did very high coverage. We did 80x sequencing as opposed to 20 to 30x for germline. We wanted to be sure we were not missing something, and to be able to, we also did SNP chips to be able to look at copy number. We had a multi-step calling algorithms and manual inspection of all the putative events. The design of our study was published not so long ago in the American Journal of Epidemiology, as shown here. We've known that all along that there are de novo mutations in families. So building block of evolution is that every generation there are these errors that take place in the ova and sperm of so to speak, the parents that are post-zygotic mutations that lead to de novo mutations. It can be 40, 60, 70, or whatever. And both the Icelanders and here in Virginia, across the bridge here from us, very strong evidence that there was an increase with paternal age, okay? So an essential type of mutation is the de novo mutation. It's due to random mutations in the gametes, the sperm and oocytes, and a small number of variants are generated. Their transmitted variants are known as de novo mutations. In other words, but they're also Mendelian errors because you don't see the mutation in the mother or father of what's seen in the child. They're the only class of genomic variation to not undergoing purifying selection. And the DNMs account for a fraction of neurodevelopmental disorders like autism, catastrophic pediatric diseases, and the like. Oh, sorry, I just need to go back one there. And they're ascertained by analysis of both parents with the children, seen only in children, but not in the parents. There have been animal data that suggested with very acute exposures, possible transgenerational effects. The evidence of double-strand breaks and non-homologous end joining was clearly there in large structural and chromosomal events, but not in the single base mutational signatures. There have been human studies of looking at the lower protracted doses, small underpowered studies, microsatellites, people looking, Alec Jeffries and Yuri Dubrova published this back in 1996 of 15 microsatellites, suggesting an increase in microsatellite errors in children, particularly in the Belarus and the Ukraine. How many of those were actually de novos or not is an open question. And then in the Nagasaki study of 3 families were anecdotal and really showed no substantial or scary evidence per se of, although it was very small, of de novo mutations. So again, we had extensive commitment to the radiation exposure. So here is a paper that was published not so long ago, the gonadal doses of this particular trio study, where we could, with exquisite detail, try and estimate what we maintain is the actual exposure to radiation in individuals in the liquidator cohort. Our principle for detecting de novo mutations was to do maternal and paternal sequencing and the child sequencing, look at variants inherited from the mother, which you could clearly see in the father, and that's the vast majority. Then you had these Mendelian inconsistency errors, not in either parent, a small but real number. Most of them were sequencing errors, were filtered because we had high depth and quality probabilities. We could go back and use the paradigms that Augie Kong for decode and Wendy Wong in Northern Virginia had used. And then we actually manually inspected every possible Mendelian inconsistency error and had very rigorous rules that are published in our supplements to our science paper. We had the power to be able to look and observe mother-father radiation doses and ask this question, an 80% power to detect a radiation dose of 0.6 DNMs per 100 milligray, or 5.5 for the mothers, which is telling us that the distribution, we had more fathers who had higher doses than mothers who had higher doses, and that's not unexpected considering the vast majority of the liquidators were men and not women. So when we looked at the association of age at conception, cumulative radiation dose, and smoking history, we could see that, for instance, maternal age, there was a borderline suggestive finding of maternal age. The paternal age was very strong, as expected. If we didn't see this, we would have worried about the structure of our study. When we looked at the cumulative radiation dose, we saw no effect whatsoever. In fact, the estimates were in the other direction, but the p-values were really clearly null. We looked at smoking in maternal and paternal, and we were very excited originally about this maternal current smoker, but when we went to some of the other large studies and asked them, they clearly squashed that finding. As you can imagine, had there been really strong evidence for smoking in DNM, that would be a tremendous public health statement, that public health finding that would need to be really investigated, but we could not see that in the larger studies. All right, so when we then looked at the parent of origin, so this was in 42% of the de novo mutations, we could look at adjacent SNPs and be able to figure out which chromosome it actually fell on. Was it the maternal or the paternal? There, you could clearly see that the dose was not showing us anything in either of these. But if you looked at the age, the father was still very strong, 10 to the minus 13th. And the mother, the maternal age became stronger. And this is something that was more congruent with some of the more recent studies from Decode and the like. It's a smaller effect size and a smaller fraction, but clearly, the old adage that when people are, women or young girls are born, that there's very little in the way of turnover is probably not completely true. So we looked at sensitivity analyses of the impact of maternal and paternal cumulative radiation dose modeling on association with DNMs, looking at the dose, whether it was the cumulative or whether it was truncated, a cumulative log radiation dose. We tried every possible way to look at this and come up with it. And we did assessment of modeling approaches on the association between dose and DNMs, linear model robust variance, Poisson models, to try and identify if we could see any real signal, and we couldn't. We know that there are many different types of events. Certainly microsatellites, mini satellites make up about 3%. Sign events, DNA transposons and the like. These are the kinds of repetitive DNAs where there's always this hypothesis of slippage per se. So when we look closely at all these different elements, we could not see it. So when we got to the estimates of the cumulative radiation dose, so for instance, here shows when we actually look by looking at the total, you've already seen those numbers. The SNVs, the single base changes, we saw nothing significant there. The microsatellites, we didn't see anything. The insertion deletions, we didn't see any. And complex and clusters have these rare events where there may be 3 SNVs and a small deletion all within a given space, first defined by the Icelanders. And these things are exceedingly rare. So in 130 individuals, we saw an average of maybe 2 per individual, even less than 2 if you put the complex and clusters together. And we didn't see any unusual distribution of those by radiation in any particular way. So the de novo signatures by preconception ionizing radiation dose, you could see we're really no different by radiation doses here, by quartiles of radiation dose. When we compare them to the studies of Iceland and Northern Virginia and other studies, we saw that the distribution of the single nucleotide changes were absolutely unchanged, whether they were in CPG islands or whether they were non-CPG. In other words, if they were in chromatin accessible versus non-accessible regions per se. One of the things that a couple of colleagues and people have been writing and wanting to say, oh, well, the background is higher with respect to the absolute number of events in Ukraine versus these others. Yes, that is true. It's like 12 versus 16 microsatellites, but I would caution you that these are not fixed final things in the sense of the bioinformatic programs are accurate, but not to 100% degree. And so if someone is going to compare our data to the Icelanders of Northern Virginia, they need to get the original BAM files and run all the samples exactly through the same pipeline, because that little difference may be just enough to say 12 versus 16. And again, these are tiny numbers compared to what we see. So we don't see any real material difference between the Chernobyl trios and the population-based studies. Now, our study is an evaluation of peripheral blood. There's a survivorship bias here, and we knew that that was the case. At the time of the accident, 1986-87, there clearly were catastrophic events that were taking place. There were in utero exposures. Those were not captured. Those were not cataloged in any meaningful way so that scientifically we can ever really understand those. But when we look at individuals who were born more than a year, there was no increase in DNMs in children born in 1987 in any substantive way in any of those categories. And the question is, if someone had two more microsatellites, the statistical likelihood that those are in particular regions that are going to cause disease is exceedingly small. We have twins that we now know that we can analyze, and we're looking forward to doing that in the coming year. We were unable to investigate really acute high doses in the immediate aftermath, as I mentioned. Although we did short read sequencing technology, we had SNP chip analysis. So we're able to combine those. We probably didn't miss many, if at all, large events. We are doing the long range Oxford nanopore and PacBio on a small handful to be absolutely sure. So in our transgenerational study, just want to say that we think this is of great public health importance. There's adequate power to identify substantive increases in DNMs in adult children born to liquidators. And this is something that we think could be of real value to other places in the world, such as Fukushima, where we've had quite a bit of press since this paper came out in Japan, in assuaging individuals that where one of the major reasons people were not moving back to Fukushima was concern about transgenerational effects. Our doses were both extended and lower than animal studies. So there could have been lower rates of gonadal DNM events. And this raised this interesting question of the balance between new gonadal DNMs and possible DNA repair. So contrast this with the high dose burst of energy that's seen with radiation therapy or in very acute high doses as used in many of the animal studies, as opposed to the liquidators like the children that I talked about in the first study, where there was a protracted exposure to background radiation as opposed to getting a major burst per se. We saw no evidence of radiation-induced single-base or epigenetic signature. I didn't show you the epigenetics, but those are completely null. Adequate power to identify substantive increases. And we are doing further studies looking in some of the lower dose in Chernobyl, but we've had a long-standing collaboration with our colleagues at the Radiation Epidemiology Research Foundation in Hiroshima, and talking about looking at the trios in the surviving families of the Hiroshima and Nagasaki blast. Same time, we know that clonal hematopoiesis and genetic mosaicism can be increased with radiation therapy, and we're just completing those more detailed genetic analysis, targeted therapy, targeted sequencing, and hopefully later this summer we'll start reporting on that. So let me recognize particularly Meredith Yeager and Mitch Makila, who were really instrumental in the genetics of this, with Amy Barrington, the epidemiologist who really was critical for the design of this. And of course, our collaboration with Dmitry Bazika, academician Bazika in the Ukraine, and certainly our colleagues at the Broad and elsewhere have led to this. So with that, I'd like to thank you for your attention today, and for all those trainees out there. The Division of Cancer Epidemiology and Genetics that I direct is a marvelous place. We have up to 130 trainees at any one time, and we do many different things. Here are just some of the kinds of studies, and we are open and seeking terrific individuals for further training and academic development with our very rich data sets. And so with that, why don't I stop and see if there are any questions?
Thanks, Stephen. That was fantastic talk. Very, very interesting. Definitely quite surprising that radiation doesn't seem to increase the rate of de novo variants. Something I would have expected otherwise before seeing your paper. If anyone would like to ask a question, could you please raise your hand in the chat? In the meantime, I have a question to ask. I kind of read in an online interview that you did with Science Magazine that you're hoping to extend the study to include more children and grandchildren of the exposed individuals. Yeah. Given the lack of evidence for increased de novo variants in the kind of generation 2, do you think there's still much of a rationale to look at the third generation?
Well, the third generation is an interesting epigenetic question, as you know from all of the data about starvation and nutrition after the Second World War, in the famous Dutch and the Danish studies. So with that there, we feel we have an obligation to look at that at some point. I think, again, this question of the lower dose versus higher dose is very important. And we're not going to sequence necessarily quite as many as we did before, but we are going to be looking at those that we still have access to who had higher dose, and the twins. I'm really intrigued by the twins. To me, we don't know just from the history whether they're monozygotic or dizygotic, and that we're playing not a game, but with our colleagues, we'll let the genetics tell us that. I mean, it'll be obvious in seconds when we start lining those things up. But we really strongly think that will be a very interesting place to look at this kind of question.
Thank you. I don't think there seem to be too many other questions in the chat that I can see. I guess that means I get to ask another question then. I'm curious if there's been much research into kind of the height of children who are exposed to radiation, or the the height of children where their parents were exposed to radiation. Do you see much there? Because obviously height is very much affected by negative environmental exposures.
Yeah, well, that's an interesting question. We're just sort of getting some of these other kinds of studies launched. And now that, for instance, from the Chernobyl, you have all of these people who have clearly attained their adult status. I mean, the event was 35 years ago. So height and weight would be a very interesting thing to study at this point and see what's radiation exposure related to that. And so we have some colleagues who are looking at some of those kinds of questions.
Great, thank you. As I can't see any other questions, I'd like to thank Stephen again for presenting. Super interesting stuff. Yeah.
Well, thank you. And it's a pleasure talking to you all. And I look forward to seeing you all in person, I hope in 2022. And if George is on, I hope that we'll get to meet Bruno again. I went to the first conference in the Mendel Monastery, and that was quite memorable and quite remarkable. Who knows, we may actually have some MR for this study. All right, thank you. All right.
Yeah, last thing was, I think George said something in the chat. He said they'll be inviting, he'll be inviting you to the next one.
All right. Thank you, George. All right, good. Well, it's nice to see you all. Stay well, and we will see you all in 22, okay?
Thanks all, bye.
All right, bye-bye.